If you thought the human genome was complicated, you're going to love proteomics. It takes genetics to a whole new level. You know those 30,000 genes that are always called a blueprint? Haven't you ever wondered, a blueprint for what? The answer is proteins. Our genes provide the recipe for the hundreds of thousands of proteins in our bodies. And it's proteins that do all the work. They regulate metabolism, enable us to move and breathe. They help build our hair, skin, and bones. But mapping the human proteome, as it's called, is so complex. It's said to hold 1,000 times more information than the genome. And scientists are very keen to chart these unknown waters. At this workshop in Windsor, they spend hours navigating a single protein on their laptops.
There is a very finite universe of protein structures.
Dr. David Wishart is their guide and is one of the leading proteomics researchers in the country.
I guess you could say that proteins are the engines of life. And they're amazing machines. They can self-assemble. They're nanomachines. They are measured in the size of nanometers. And they can do many strange and wonderful functions, from repairing damaged tissue to assembling to make little motors, which you'll find in the flagella of bacteria, to pumps, to valves, to crawlers, to stepper motors. Proteins are able to do all of this, and that's because of the three-dimensional shape they adhere to, and that's all determined by their sequence.
In the world of proteins, sequence is everything. Proteins are made up of amino acids. There are only about 20 amino acids in the body, so how complicated could it be, right? Well, when you consider that there can be hundreds, even thousands of amino acids in one single protein, the picture becomes a little more blurry. Then there's the problem of structure. Proteins don't form a nice chain like DNA. They fold and bend and do all sorts of contortions to form a unique shape. And in the world of proteins, shape is everything too.
There's a path that you go from an unfolded protein to a folded protein. And somehow
Dr. Chris Hogue talks a lot about the fold, shape, and architecture of proteins. At times, he sounds more like an engineer than a biologist. That's because the protein's structure literally determines the job it will do. More specifically, what other proteins it will bind to.
The key issue is that understanding the three-dimensional shape, it's that three-dimensional shape, how the protein sequence folds into a three-dimensional shape that gives you the function, that gives you the binding surfaces. When I define function as the set of interactions, you can't have that set of interactions until you have the shape.
At the Samuel Lunenfeld Research Institute in downtown Toronto, Dr. Hogue and a team of scientists have been quietly investigating proteins for the past 4 years. Like many in the field, Dr. Hogue is very comfortable with computers. There are 108 in this room, processing thousands of bits of data. One area of study has been the yeast cell. Because of its similarity to many human cells, the proteome of the yeast cell has been probed in detail. Lots of detail. 600 proteins have been identified and more than 4,000 interactions between proteins. And you thought the yeast cell was a simple organism. All these interactions are as important as the proteins themselves. One single protein fitting nicely into another can trigger a cascade of events. It's a chain reaction so delicate that in humans, if it's interrupted at any stage, it can mean the difference between fighting off a disease and causing one, like cancer. The protein hemoglobin, which carries oxygen in our blood, is a great example of how proteins should work.
So this is hemoglobin. It's quite well designed for transporting oxygen. Each of these 4 subunits can bind oxygen. Each one can bind a separate molecule of oxygen. So each heme ring here hooks onto an oxygen, and so it packs a lot of oxygen in one protein molecule.
Today we know the function of about 20% of human proteins, but almost daily new ones are being discovered. So to store the massive amounts of information, the research center led by Gary Bader has set up an elaborate database called BIND. It's destined to become the largest proteomics database in the world.
So I type in GRAB2 into the BIND text query. This protein is a protein involved in signal transduction in the immune system. And if I wanted to find out quickly what all the other proteins that GRAB2 interacts with, it queries the database and shows me everything in the database that GRAB2 binds to. All of these boxes represent proteins just like GRAB2. And it gives me a better idea of how GRAB2 fits into the network in the cell.
Scientists from around the world can log on and find out essential details about the protein they're investigating. That will hopefully speed up research and eventually lead to the holy grail in proteomics, curing disease. That's where companies like MDS enter the picture. Barely a year old, it's part of a new wave of proteomics companies betting that proteins will pay off. They hope to find the protein culprits, ones that trigger disease, such as breast and colon cancers. Using cells taken from actual patients, researchers identify clusters of proteins they suspect play a part in the disease. The proteins are purified, harvested, blasted with electricity, all in an effort to isolate them and determine what they're doing in the body. So complex is their investigation that their bank of computers is the fourth most powerful in the world. Mike Moran is the senior scientist at MDS.
Proteomics offers just a whole other level of approaches to study disease, a whole new arsenal of equipment and technologies to discover new medicines. And these technologies in proteomics are much more geared towards actually discovering drugs than I think genomics is, because drugs don't act on genes, they act on proteins, and that's what is being discovered in proteomics. What's really important to understand is that drugs work because they bind proteins. That's in fact what drugs do. Their primary target is they're designed to have ideally one specific protein they bind to, and hopefully it's the protein that's gone wrong and is causing a disease.
It's attacking disease on a molecular level, which could lead to enormous benefits, like reducing the time it takes to research a drug, cutting back on animal testing, and minimizing side effects in the patient, since it's only the faulty protein that's targeted, not healthy cells. One drug is already being held up as a poster child for proteomics. It's called Gleevec and was approved in Canada last fall. It's for patients with chronic myeloid leukemia, a deadly cancer caused by a mix-up in DNA information. The result is a flawed protein called BCR-ABL, which signals the cell to reproduce over and over again, causing the cancer. But Gleevec moves in like a molecular missile, blocking the signal and disabling the protein. It's proteomics at its best. Gleevec showed a startling 90% success rate in clinical trials, and its sales are climbing fast. That's why the researchers at MDS are so optimistic. Their target is to develop 1,000 potential drug therapies in the next 5 years. But targeting proteins is not new. The thousands of drugs on the market today target about 500 proteins. What's different now with proteomics is the pace of discovery. Computers can process protein sequences, their folds, their interactions, in a matter of seconds. Where a single protein once took a lifetime to discover, hundreds of thousands will be determined in the next 10 to 20 years. And along with them will be drugs, neatly fitting into that molecular matrix, making sure the proteins behave.
Right now, we have a lot of strong drugs that take out one or two targets, and we may move to a world where we have a whole bunch of weak drugs that are really quite non-toxic, but in the right combination, they take out enough targets that they will stop a cancer or change the way a disease is progressing.
We probably will have thousands of new targets, and what's more, many of the drugs of tomorrow won't be the small molecules to stop the proteins. We'll actually look at the body as a pharmacy in itself, that the drugs of tomorrow will be the proteins that our body already produces to keep itself healthy for most of our lives.
Some say that the laboratories of today will become obsolete. Wet labs with glass and microscopes will give way to this. Scientists with 3D goggles building what looks like molecular Lego. But the complexity of proteomics belies a very simple goal: to truly tackle disease at its source.